Hello dear students, in this video you're going to discuss a bacteria that is a very important topic, Staphylococcus. So from the name itself, Staphylococcus, it's a cocci. So the previous year questions which has been asked from Staphylococcus, we had a question just as Staphylococcus aureus, a 5-mark question. So you have to write all the things which you know about Staphylococcus. Then staphylococcal food poisoning was asked. Then antigens, toxins and enzymes of Staphylococcus. So these were repeatedly asked questions from the university question papers. So we are going to classify Staphylococcus first before starting to learn. Like Staphylococcus, it's a genus name. There are different species under this which are causing disease, which are pathogenic, which are not pathogenic. So what you need to learn is just the important species, the medically important species. So the most important Staphylococcus species is Staphylococcus aureus. That is the most important, most pathogenic staphylococcal species. Other than that, you have two coagulase negative staphylococci. So there is a test called coagulase test. It's an enzyme produced by this bacteria. So based on that enzyme, you classify this into coagulase positive and coagulase negative. Staphylococcus aureus is coagulase positive staphylococci, and the others which are coagulase negative, they are Staphylococcus epidermidis. Like the name itself indicates, it's present in the epidermis of the skin. So it's usually a normal skin flora, but rarely it can cause diseases. And you have Staphylococcal saprophyticus. Saprophyticus, the name means it's present everywhere, like it's present on all the dead and decaying tissues and all. So it is also a ubiquitous, it's also a normal flora like species, but rarely that can also cause diseases. So the most pathogenic staphylococci, if anybody asks you, that's Staphylococcus aureus. Aureus, the name, it's because it produces a golden yellow color pigment. So what are the infections that Staphylococcus produce? In single term, you can say it is pyogenic infection. So what is meant by pyogenic? Pus forming infection. So all the pus forming infections, when you encounter a pus forming infection, the first bacteria that should come to your mind should be Staphylococcus aureus. And you can see the morphology of Staphylococci here. How does it look like? It looks like a grape-like clusters, a group of cocci which look like a grape-like clusters. So they are gram-positive cocci around 1 micrometer in size, which look like grape-like clusters. And they are aerobes basically, but they can also grow in the absence of oxygen. So you can call it as aerobes and facultative anaerobes. They can grow on all the ordinary culture media. Please note that they can grow on all the ordinary culture media. They don't have much of specific requirements. They can grow even in nutrient agar. And the optimum temperature for their growth is 37 degrees Celsius. That is the most most good temperature for most of the bacteria. 37 is the human body temperature. Coming to the pathogenesis of staphylococci, so you should know how does staphylococci cause infection. For that, what all things do they have? So you might be remembering what we have learned in the introductory classes, virulence factors. Those are the virulence factors are the things which bacteria possess in their structure or the things which they produce which helps them in causing diseases. Okay, so for staphylococci, you can classify the virulence factors into 3. They have some antigens which are present in their structure, in the cell wall. They have some enzymes which they produce, and they have some toxins. So this was a very important previous year question. Antigens, enzymes and toxins of staphylococci. Coming to the antigens, those are the structural antigens. Structural antigens means antigens which are present in the bacterial cell wall itself. So when you see the bacterial cell wall, you can see this is the cytoplasm, you have the cytoplasmic membrane, then comes the cell wall. Cell wall has got different layers. The most important layer is the peptidoglycan layer. You know that is the most thicker layer in gram-positive bacteria. In addition to that, it has a teichoic acid layer that is also specific for gram-positive bacteria. So that is there. In addition to that, it has got certain cell wall protein antigens and capsule also in some strain. Very few strains of staphylococci possess capsule. So what is more important is peptidoglycan, teichoic acid and a special protein which it has got called protein A. This helps in this protein A, it can bind to the immunoglobulin, Fc portion of immunoglobulin. Immunoglobulin, if it's a Y-shaped immunoglobulin, you have an The lower end is called the FC portion that can be bound to this protein A. So protein A of Staphylococci is responsible for a co-agglutination reaction. It's a kind of agglutination reaction by binding to the FC portion of the immunoglobulin. So protein A is responsible for the co-agglutination. Probably it can be an MCQ question, but not that important in your theory part. And in addition to these capsule, this peptidoglycan, teichoic acid, protein A and all, Staphylococci has got a clumping factor. It's called a cell surface adhesin. Adhesin means something which will adhere to the host cells, which will adhere to something. So this clumping factor or this adhesin helps in adhering to the host cell, and also it helps in clumping of the cocci together. When you put staphylococci along with some plasma, they get clumped together. This is because of the cell surface adhesin. We'll come to that later. So those are the antigens. Coming to the toxins which staphylococci possess, you have hemolysins with which it will lyse the RBCs. It will produce hemolysis on blood agar. Then leukocidins are there, enterotoxin, exfoliative toxin, and toxic shock syndrome toxin. Please note the last three: enterotoxin, exfoliative toxin, and toxic shock syndrome toxin. These three causes the toxin mediated diseases. These are responsible for the toxin mediated diseases of staphylococci. So we can make a mnemonic for this, remembering the toxins of staphylococci: HEAL T, hemolysin, enterotoxin, exfoliative toxin, leukocidin, and toxic shock syndrome toxin. Coming to the hemolysins, staph has got 4 types of hemolysins: alpha, beta, gamma, and delta. Okay, and alpha hemolysin, it's very cytotoxic. It will destroy WBCs, platelets and all. Beta hemolysin, it will act on the sphingomyelin present on RBC membrane. It's called as sphingomyelinase. So that is responsible for the hemolysis on blood agar. Then you have gamma and delta. And other than hemolysin, they have a leukocidin as such, which can lyse, which can destroy cytotoxic to the WBCs, leukocytes. It is also called as Panton-Valentine toxin. It has got some synergistic action along with this gamma hemolysin. And they can damage neutrophils, they can damage macrophages, so they are very cytotoxic. So all these hemolysins and leukocidins helps in causing cell damage. So that can lead to inflammatory reaction, tissue damage, necrosis, and all the kind of pyogenic infections which it forms. So those helps in establishing infection and causing tissue damage in staphylococci. And what are the enzymes they possess? So we have finished antigens, we have seen what are the toxins. The next thing is what are the enzymes. The most important enzyme that Staphylococcus possesses is the coagulase enzyme. Coagulase enzyme is present, I told you it's present only in Staphylococcus aureus, and in others it is absent. So other than coagulase, we'll see what is coagulase enzyme. It's important, you have to see a bit of it. Phosphatase and deoxyribonuclease are the other enzymes which it has. Then lipase, hyaluronidase and proteinase. See, all these will act on the cellular components, on the phosphates, on the phosphate membranes, DNA, lipid membranes, then hyaluronic acid which is present in the joints, synovium, protein molecules. So all these will cause cell membrane damage, nucleic acids will be lysed, lipid membranes damaged. So everything will help in the pathogenesis of this organism. So basically coagulase is the thing you should not forget, but also remember it has got different enzymes which can break down different molecules present in the cells. Coming to coagulase in a bit detail, because this is the most important enzyme, there are 8 types of coagulase enzyme, A to H, of which coagulase type A is the one which is produced by the human staphylococci. And what does coagulase do is it can convert fibrinogen to fibrin, and they can lead to formation of a clot or a coagulum. That is why it's called coagulase. It forms fibrin and thus it forms a clot. And you have 2 kind of tests for coagulase enzyme. One is a slide test and a tube coagulase test. We'll see what it is. Free coagulase, that is when you say about coagulase enzyme, what you mean is the free coagulase. Hope you remember we just mentioned about a cell surface adhesin or a clumping factor. That is called as the bound coagulase enzyme. So slide coagulase test is for detection of the bound coagulase, and tube coagulase test is for detection of a free coagulase. So slide is for bound and tube is for free. Okay, and what do this free coagulase do is, free coagulase enzyme will combine with a plasma protein that's called as coagulase reacting factor, CRF. They together will activate prothrombin and they will convert fibrinogen to fibrin. That is how they form fibrin. That is coagulase plus CRF will activate prothrombin, converting fibrinogen to fibrin. Thus they form a clot. Hope you can see in this figure. You can see a tube, you can see a coagulum formed here. So you add plasma, you add staphylococci to it, and you incubate it for some hours. When you take it back, you can see there is a clot formation, a coagulum formation. That is the principle of tube coagulase test. Whereas slide coagulase, what you do is you take an emulsion of staphylococci and you add plasma to the slide. What happens is the staphylococci cell wall contain the cell surface adhesin or the bound coagulase, which will bind to the fibrinogen in plasma, leading to clumping of cocci. So all the cocci will get clumped together. So you can see some kind of white particles clumping together. That is the bound coagulase, due to presence of bound coagulase, and this test is called the slide coagulase test. Basically, we do a slide coagulase in a slide and tube coagulase in a tube. So tube is for detection of T4F. Tube is for detection of free coagulase and bound coagulase is for detection of slide coagulase. So coming to this diseases which staphylococci produce, I told you it's everything about pyogenic. So abscess, a local pus collection. If you see an abscess case, then the most important organism associated is staphylococci. So in this figure, you can see this bacteria is multiplying here. It is invading into the cells. It's producing the toxins. It's releasing the enzymes, causing cell damage. It will attract all the inflammatory cells to this area. And due to formation of this coagulase enzyme and formation of a clot, it has a tendency to form localized infection. So the infection will be a very localized form that is called as abscess. So if you see an abscess, think about staph first. Okay, so I have a mnemonic for you to remember the infections produced by staphylococci, a softer lens. So A for abscess, I don't want you to forget abscess, that is why I put A. Then what is S? S is for all the skin infections that staphylococci cause. It can be carbuncle, furuncle, impetigo and all. And O for the ortho things, that is osteomyelitis, arthritis. Okay, and F for food poisoning, T for the toxic shock syndrome toxin, and E for endocarditis. R is for the renal things. Renal means it can cause UTI, it can cause pyelonephritis. And L is everything related to lungs, lung abscess, empyema formation. And again you have this E, one E is exfoliative and the other E is endocarditis. N is for necrotizing fasciitis and S is for sepsis. So these are the infections caused by Staphylococcus. Suppose you get a 2-mark question, the mnemonic may help you. Abscess, skin infections, osteomyelitis, arthritis, food poisoning, toxic shock syndrome, exfoliative skin syndrome, the renal problems, the lung problems, endocarditis, necrotizing fasciitis, and sepsis. So all these are the infections caused by Staphylococcus. When you see the cutaneous infections, you can see it can be simply a pustule, just a pus formation. It can lead to multiple hair follicle involvement and crusting of these multiple follicles together, folliculitis, that is called the carbuncle. So when a single hair follicle is involved, you can call it as a folliculitis. When there is multiple folliculitis, involvement of multiple follicles, you call it as carbuncle. And it can be impetigo. You can see the crusted lesions, that is impetigo. This is an abscess formation. Addition to this, staphylococci can cause wound infection, surgical site infection, burns infection, and so on. And when somebody asks you what are the deep infections caused by staphylococci, remember the lung infection, renal problems, osteomyelitis, endocarditis, and sepsis. So, staphylococcal food poisoning is due to a preformed enterotoxin in food. Suppose the food is contaminated, food is containing an enterotoxin produced by staphylococci, it can lead to the symptoms of food poisoning within 2 to 6 hours. So that is staphylococcal food poisoning. So you have an enterotoxin produced by staphylococci. It's a super antigen. It's a very important antigen, super antigen. It is usually seen to contaminate milk products and meat products. So when staphylococci enters into a food item like a milk product or a processed meat, it will release this enterotoxin into the food. So if you consume the food which is already containing this toxin, you will get the symptoms of staphylococcal food poisoning. And the symptoms will be like nausea, vomiting, or diarrhea. And usually you get the symptoms within 2 to 6 hours. This time period is very important. Most of the food poisoning happens little late. But staphylococcal food poisoning is very fast. You get the symptoms within 2 to 6 hours. This is because of this, remember, preformed enterotoxin. And staph enterotoxin is a super antigen. Don't forget that. Now, staphylococcal scalded skin syndrome, it's due to this particular toxin called epidermolytic or exfoliative toxin. And it will be like there are two types, A and B. Initially, there will be tender blisters and bullae formation, and later on, the epidermis will be getting separated or peeled off. That is why it's called exfoliative. And it's known as Ritter's disease in newborn. In adults, you call it as toxic epidermal necrolysis. That is the two terms we use to describe staphylococcal scalded skin syndrome or exfoliation. Now the next toxin associated syndrome of staphylococci is toxic shock syndrome. This is due to a particular toxin that staphylococci produce. You remember the toxin HEAL T. T is for toxic shock syndrome toxin. So this toxin is responsible for this particular syndrome, and the symptoms will be like the patient will present with fever, rash, hypotension, myalgia, vomiting, and finally leading to shock, confusion and all. So there will be multiple organ involvement because this toxin is also a super antigen. So it can lead to multiple system involvement like liver, kidney, GIT and CNS. And this was mostly seen in women using vaginal tampons. So it was like before there was a tendency of using more use of vaginal tampons, and this toxic shock syndrome was seen in those group of women. And now this has come down a lot because it's not being used nowadays. So that's why it's not that common now. But still, you have to know about toxic shock syndrome. It can happen even as part of any other infection, not just because of use of vaginal tampons. So it's due to this toxic shock syndrome toxin, which is a super antigen. Coming to the lab diagnosis, Staphylococcus. So, you know, what all specimens will you collect? For any bacterial lab diagnosis, you have to know what are the specimens that you take, what microscopic test you do, what culture you do, and how you identify it, and finally, how you will get the antibiotic sensitivity. So, specimen will depend upon the type of disease it produces. If it's an abscess, you have to take the pus, respiratory infection, sputum, UTI, urine, or if it is a sepsis, you have to get blood, like that. It differs. If it's a food poisoning, you have to go and collect the stool sample, right? Or you have to take the food sample or the vomit sample. So, like that, the different specimens varies. You have to collect the specimen in sterile containers under aseptic conditions. If you're taking pus, you have to get it in sterile swab. Always in any kind of culture, sterile swab, sterile container and aseptic precautions are very important. Now, what do you do for microscopy with the sample? You do a gram staining. You remember gram staining. And how will you see that? You see like this, gram positive cocci in clusters along with pus cells. Right? So that is the staphylococci. And what will you do for culture? You can culture it in all the medium, including nutrient agar, blood agar, and McConkey agar. And you have some special selective media for staph like mannitol salt agar, salt milk agar, and Ludlam's medium. So at least remember one selective medium for Staphylococcus. I prefer mannitol salt agar. That's the best medium. You incubate these medium at 37 degrees Celsius for 18 to 24 hours. And what will you see in the plates? If it's a blood agar plate, you'll be able to see the beta hemolytic colonies. I told you it produces a beta hemolysin, alpha, beta, gamma, and delta. Beta hemolysin is responsible for the hemolysis it shows on blood agar. If you examine the nutrient agar, you can see very beautiful golden yellow color colonies. This is because Staphylococcus produces staphyloxanthin pigment, which is golden yellow in color. That's why nutrient agar, you can see the beautiful golden yellow color colonies. And how will you identify these colonies and confirm it is Staphylococcus aureus? Again, you can do a gram staining test. And additionally, you can do some other test. What other test will we do? You do a catalase test. Remember, catalase test is positive in all Staphylococci, not just aureus. Okay, if catalase test is negative, you have to put that under streptococci. So this is catalase positive. And what about coagulase test? I told you coagulase test is positive only in Staphylococcus aureus. If coagulase test is negative, it's not aureus, it can be some other staphylococci like epidermidis or saprophyticus. You have some additional tests like mannitol fermentation. gelatin liquefaction, and DNA test. All these are positive only for Staphylococcus aureus. Even if you forget these tests, please don't forget coagulase test. And finally, you perform an antibiotic sensitivity testing. With that, you find which antibiotics are useful for this particular bacteria. So once more, I'm showing the coagulase test, tube for pre-coagulase and slide for bound coagulase test. I hope you understand the principle. Okay. Now coming to treatment, how will you treat a staphylococcal infection? This is very common. You will definitely encounter staph infection in your practice. Penicillin, if sensitive, that is the best drug, pensyl penicillin. But there is very high level of resistance to penicillin. So then you go for cloxacillin. Cloxacillin is used for beta-lactamase producing strains. Suppose if it is even resistant to cloxacillin, then you call that strain as methicillin resistant Staph aureus. Then you have to go for very higher end drugs like vancomycin, linezolid and teicoplanin. If you have a mild superficial lesion, you can just apply topical antibiotics like bacitracin or chlorhexidine is enough. Antiseptics or antibiotics is enough for skin lesions where you don't have to go for a systemic antibiotic. So remember penicillin, if penicillin resistant, cloxacillin, if cloxacillin resistant, vancomycin or linezolid. Then just a word about the antibiotic resistance mechanism of Staphylococcus. I told you it can be resistant to penicillin. Why it is resistant to penicillin? Because they can produce penicillinase enzyme, or it's a beta-lactamase. Penicillin is a beta-lactam compound. If it has got an enzyme to lyse the beta-lactam compound, then it can easily destroy the drug. So it's coded by plasmid. Penicillin resistance in Staphylococcus is plasmid coded. Hope you remember the genetics we learned. How can they transfer the resistance through plasmids? It can be transferred by transduction or also by conjugation. Okay. Then there is another property, another mechanism by which it becomes resistant to cloxacillin also. It is because of reduction in the affinity of the penicillin binding protein. This bacteria has got a special protein called penicillin binding protein to which the penicillin molecule acts. If this protein becomes less affinity becomes less for this penicillin binding protein, penicillin cannot act on it, cannot bind on it. So in that case, it becomes resistant even to cloxacillin. So reduction in the affinity of the penicillin binding protein, which is coded by a special gene called MEK-A gene. Suppose you get a question on MRSA, methicillin resistant Staphylococcus aureus, please write it is MEK-A gene coded and it's chromosome mediated. It's coded by a gene called MEK-A gene. And what will happen? It will lead to reduction in the affinity of the penicillin binding proteins, leading to MRSA. So if it is MRSA, you have to treat the patient with vancomycin or linezolid. So I'm repeating the previous year questions. Staphylococcus aureus as such. Then please write about the morphology, about the virulence factors, diseases, lab diagnosis and treatment. If it is food poisoning, just write about food poisoning. Remember the preformed enterotoxin and the incubation period of 2 to 6 hours. And the antigens, toxins and enzymes of Staphylococcus. That's also very important. Thank you.